Today I will talk about the human cardiac stromal cell, which we've been working for a few years now. I will present you some data which have already been published, and then some more recent data which is still unpublished. So when we started working with these cells, what we knew is that bone marrow mesenchymal cell transplantation into the infarcted heart improves left ventricular function and remodeling. And we also knew that tissue-specific cells, in general, might be better for cardiac repair than cells from other sources. So we wanted to isolate, culture, and characterize human cardiac stromal cells and compare these cells' properties in vitro and vivo to those of bone marrow-derived cells from the same patient. So we utilized cells from patients undergoing cardiac surgeries. The cells were obtained, the cardiac cells were obtained from the right atrium, from the specimen which is obtained at the time patients undergo cardiac surgery. Since patients also have sternotomy in these type of procedures, we obtained bone marrow mesenchymal cells from the sternum, from the sternal blood of these patients. We isolated the cells and then we cultured them under similar conditions. And so we were able to compare human mesenchymal cells, stromal cells from the same patient, from the heart and the bone marrow. The stromal cells from the heart and from the bone marrow looked alike. They expressed the usual mesenchymal markers. There was a significant difference in CD90 expression. Around 85% of the cells from the bone marrow expressed CD90, whereas only 30–35% of the cells from the heart, stromal cells from the heart, expressed this marker. That was the only significant difference, in addition to the expression for c-KIT. Here we're looking at a number of pluripotent and stemness-related genes. They're essentially alike in the cells from the heart and from the bone marrow, with the exception of c-KIT, which is more highly expressed in the cardiac cells. Then we looked at the early and late cardiac markers, GATA4, GATA6, KL5, MLC2A. They were all more highly expressed in the cardiac cells and in the bone marrow cells. We then wanted to look at Mirnas expressions in the two cell types. We did a microRNA profiling looking at 300 different microRNAs. Two cell types were essentially very similar, except for a few microRNAs, which clearly differentiated the cardiac stromal cells from the bone marrow stromal cells. And these are the microRNAs which helped identify the two different cell types. Now we know that the bone marrow mesenchymal cells can differentiate toward adipocytes, or they can have an osteogenic program. So we wanted to see how the different stromal cells from the heart and the bone marrow behaved in adipogenic and osteogenic medium. In adipogenic medium, cells accumulate oil red, which is an indication of fat accumulation. They accumulate fat, which is evidenced by oil red O, and the accumulation of oil red O was more significant in the bone marrow cells. So was the expression of two other indicators of adipogenesis, which is PPAR gamma 2 and adipsin. They were more significantly expressed in the bone marrow cells and in the cardiac stromal cells. When we looked at osteogenesis and we looked at alkaline phosphatase, that was largely present in the bone marrow cells in osteogenic medium, virtually absent in the cardiac stromal cells. And osteopontin, messenger RNA, was more highly expressed in bone marrow cells and in cardiac cells. So adipogenesis and osteogenesis were reduced in cardiac stromal cells compared to bone marrow stromal cells. We then attempted to make these cells differentiate over the cardiovascular lineage in vitro. And here we can see that in response to cardiogenic medium, some cardiac stromal cells express alpha-sarcomeric actin, whereas bone marrow stromal cells never did that. In these experiments, what we wanted to see was whether there was a paracrine effect of neonatal cardiomyocytes on the cardiac stromal cells. We used a transwell approach to prevent any physical contact between the two cell types. And what we found, these cells, which are cardiac stromal cells, were beating after having been cultured in a transwell in the presence of neonatal cardiomyocytes in the lower part of the well. We never observed that when we used bone marrow stromal cells in the upper part of the well. We then used a rat model of myocardial infarction and heart failure. In these experiments, the coronary artery was ligated. After 3 weeks, the animals were into heart failure. They were injected with 500,000 cells, either from the bone marrow or from cardiac stromal cells. And then after 3 weeks, they were sacrificed. This is just an example of what we found. The stromal cells were GFP labeled with an antivirus vector before injection. And here you can see 3 of these cells expressing GFP. And then you can see these cells, which also express the human Y chromosome, indicating that these are indeed the cardiac stromal cells that we had injected. We never observed this type of differentiation toward the myocardial lineage when we injected the bone marrow cells. Now, when we looked at function, we were somewhat disappointed. Here are the hemodynamic studies. Here is positive DPDT, negative DPDT. You can see that from the sham-operated animals, there is a decrease following a myocardial infarction. When we injected growth factors, IGF-1 and the hepatocyte growth factor, there was, in some cases, a small improvement. When we injected the cardiac stromal cells or the bone marrow stromal cells in conjunction with the growth factors, there was an improvement following the injection of the cardiac stromal cells, not a significant improvement in the case of bone marrow stromal cells. Again, in the left ventricular end-systolic pressure, there was an improvement following the injection of the cardiac stromal cells. No significant improvement following the injection of the bone marrow cells. No significant differences in left ventricular systolic pressure. Insofar as volumes are concerned and infarct thickness, the left ventricular volume showed evidence of reverse remodeling following injection of cardiac stromal cells. A less significant effect occurred following injection of the bone marrow cells. And again, the injection of the cardiac stromal cells enhanced the thickness of the left ventricular wall, whereas this does not occur following injection of the bone marrow cells. So altogether, this data showed that the cardiac stromal cells had a positive effect, which was relatively minor. And so we questioned whether we could enhance this regenerative potential of these cells. And so we wanted to try to reprogram the cardiac stromal cells into functionally competent cardiovascular precursors. In order to do so, we developed this epigenetic cocktail, which we call EPIC. The cells were cultured in low serum, and then they were exposed to all-trans-retinoic acid, which is a nuclear receptor activation, phenylbutyrate, which is an inhibitor of histone deacetylases, and a nitric oxide donor, which has a variety of effects, including apoptosis prevention and upregulation of some interesting microRNAs. Here you can see that the cardiac stromal cells following treatment with EPIC exhibited a decrease in HDAC activity as expected. Also, in response to EPIC, the cells stopped proliferating. There was no increase in cell death, no increase in cell senescence in comparison to control. Now here we're looking at the effect of EPIC treatment on stem cell markers. There are two stem cell markers in which we are particularly interested. Piero Anversa has identified them as key players, specifically c-KIT, as we have heard before, but also MDR1, which is described in Beltrami's paper from 2003. You can see that following EPIC treatment, both MDR1 and c-KIT increased significantly in comparison to control. Here you can see the staining for c-KIT and MDR1. And MDR1 was a channel functionally competent because rhodamin extrusion was prevented as expected by treatment with verapamil. There were other important markers that we also looked at in response to EPIC. Notch, JAGD, and NUMB, they all increased. Nucleostamin markedly decreased in response to EPIC. And here we're looking at the effect of EPIC treatment on cardiovascular and cardiac precursors. You can see that the VEGF receptor 2, GADA6, and alpha smooth muscle actin all increased in the cardiac stromal cells in response to EPIC. Some important early cardiac precursors, namely GADA4 and X2.5, increased. There was no significant effect on MEF2C. And among late cardiac markers, we found an increase in alpha-myosin heavy chain and alpha-sarcomeric actin, indicating these cells were more directed toward the myocardial and vascular lineage with EPIC treatment than before being exposed to EPIC. Regarding microRNAs, we found that two important microRNAs which have been involved in cardiomyocyte progenitor differentiation, MIR1 and MIR499, both increased remarkably in response to EPIC. We found no significant effect on MIR155 and MIR126. We then questioned whether these cells were functionally competent, and we looked at their electrophysiological properties, and these experiments were conducted in collaboration with Dario Di Francesco and Andrea Barbuti at the University of Milano. You can see that in response to EPIC, there is an increase in the expression of the gene for the fast sodium channels, so both the gene, the messenger RNA, and the protein increased. In electrophysiological studies, it was found the presence of a fast inactivating sodium current, which was TTX sensitive. And approximately 50% of the cardiac stromal cells treated with EPIC showed this type of current. It's important to say that these cells, however, were largely depolarized. The resting potential was around minus 12 millivolt, and that went along with the fact that the potassium inward rectifier, which plays a key role in maintaining the resting membrane potential, was minimally expressed. Since we did the experiments with Dario Di Francesco, we could not avoid, and we did not want to avoid, looking at IF, the pacemaker current. We found expression for HCN4, and in approximately 12% of the cells, we found evidence of IF in EPIC-treated cardiomyocytes. Since the EPIC cocktail is expected to act on the epigenetics of these cells, we looked at the epigenetic landscape, and we looked at acetylation and methylation of some key lysines on histone 3 and on histone 4. The key points here are that two events which are activation markers, indicators, which is trimethylation on lysine 4 on histone 3 was markedly increased, and so it was lysine 16 acetylation on histone 4, and these are activating markers. In contrast, two repressive markers were diminished in response to EPICs, and they are lysine 9 trimethylation on histone 3 and lysine 20 trimethylation on histone 4. We then carried out some cheap experiments, some chromatin immunoprecipitation experiments looking at promoters in which we were particularly interested in, specifically the c-KIT promoter, the NKX2.5 promoter, and the MDR1 promoter. In the case of these three promoters, we found that EPIC enhanced the activating markers. In contrast, in the nucleostamin promoter, and you will remember that nucleostamin decreased in response to EPIC, were depressed in comparison to control. So these results went along with the gene expression data that I've shown you before. So the conclusion of this work is that the cardiac stromal cells and bone marrow stromal cells exhibit distinctive properties related to their origin. Cardiac stromal cells may be epigenetically induced to acquire functionally competent cardiovascular precursor properties. Now we are in the process of establishing a even more effective EPIC cocktail and determining whether the EPIC-treated cells are more effective than their counterpart, their control, not treated with EPIC, in repairing the infarcted heart. And these experiments are ongoing. As I mentioned before, this is an ongoing work. These are the people who have collaborated to this work and have done this, carried out these experiments. I want to point out Alessandra Rossini, Matteo Vicelio, and Carlo Gaetano, who have worked very intensely in these projects. Carlo is now leaving our lab in Rome after 15 years and setting up his own lab in Frankfurt. The electrophysiology experiments have been done in collaboration with Dario Di Francesco and Andrea Barbuti, and the in vivo experiments in the rat model of myocardial infarction have been performed with Federico Quaini and his laboratory and his colleagues. And I want to thank you for the attention.